start going through. I'm just gonna I'm not gonna speed through it. I'm gonna kind of generally read. I'm gonna generally read kind of each of the different topics. If at any point any of you feel like that doesn't sound familiar, or that's confusing, or that doesn't make any sense, speak up. Or even like on Teams, you can click the little raise hand button, and we'll pause and kind of talk about it and break it down a little bit more. So, because at this point, like, we have all the videos of all the Linus questions. The Linus questions are also going to be really, really good practice. Just in, kind of practicing over and over and over again, kind of what that information. And that Quizlet is, this one's 400 something. The total is like 500 something questions. Between now, what day is your guys' exam? Tuesday?
Wednesday. Yeah.
So
and I just did the math. I still have 70 more questions to add to it.
Yeah. Give or take, that's about, if you do about 70 questions a day, that's would get through all of them. You probably don't have to do that many. Like, if you even just do 30 questions and like go decent, if you can go decently quick and hopefully remember most of it, or at least a chunk of it, you'll get through it just a little bit each day. Okay. And if I remember right, I think his exam is like 50% the newer material.
Oh.
He gave me the breakdown today. He said 15 over the last two chapters we just had, or last two things we had from the last exam. 15 to 20, he said, and then everything else is kind of sprinkled from throughout.
Okay, sounds good. Okay, let's jump in. Let's get started. Know what oxidative phosphorylation. We have to start with oxidative phosphorylation. So know what oxidative phosphorylation is and its purpose. So oxidative phosphorylation is that process in which ATP is formed as a result of the transfer electrons from NADH, FADH2 to O2 by a series of electron carriers. What's another name for oxidative phosphorylation? What's the kind of the overall process? What's that called, where oxidative phosphorylation occurs?
Electron transport chain.
Perfect. Yeah, it's the electron transport chain, right? So that location, this is going to be taking place in the mitochondria, specifically in the mitochondria. Where does this take place?
In the inner membrane.
In that inner membrane. So if you remember the mitochondria, is the powerhouse of the cell. You know that they don't say that anymore? They don't say the mitochondria is the powerhouse of the cell?
Why is that?
They use some kind of different, it's like they say it's like the pool, or I don't know, it's something weird.
Oh,
Made me very settled. Okay, so the mitochondria, remember what mitochondria is set up like this. We have an outer membrane. This is our outer membrane. We have an inner membrane. This space right here between the two membranes is called what?
The cristae?
Not quite.
The outer
The space. So it's called the intermembranous space. Because in between, it's in between the two membranes, right? Now, in the middle here, what's the middle region called?
The inner membrane?
Yeah, we have the inner membrane, so we have the outer membrane, the inner membrane. Inside of the intermembrane, this whole middle space is known as the matrix. Remember? Okay. These guys here, these loops that form. These are also called cristae. So it's part of our intermembrane. These are our cristae. He doesn't hit on that a lot like he used to. He used to make that a really big deal, so don't worry too much about that. So first, let's take a step back and think outside of the mitochondria. There's all this jelly-like stuff in the cell. What's that called?
Cytosol.
Our cytosol. What reaction takes place in the cytosol?
Glycolysis.
Yeah, we have glycolysis here. What are glycolysis? What are our products of glycolysis? What do we get from glycolysis? Let's back up a step. What goes into glycolysis and what do we get out of glycolysis?
2 ATP goes in. We get 4 out, but it's technically 2 that we actually gain.
Good.
And then NADH is what we also get out of it.
Yeah.
Yeah. How many NADHs do we get?
2.
Let's write it like this. Going in, we have 2 ATP. We have glucose. Out, we get 4 ATP. So we net 2 NADH. And what else do we get?
Pyruvate.
Pyruvate. So let's say our body, let's say we're running really hard, and we don't quite have a whole lot of oxygen available in our muscles. If we don't have oxygen available, what happens to that pyruvate?
Becomes
It turns
lactate.
into lactate.
Yeah, if we don't have oxygen, we get lactate.
Hey, Drake,
What's the-
they're in your inbox.
Okay, sweet. Thank you. What's the deal with lactate? This is going to get a little bit ahead, thinking further into physiology than more than it is biochem. But this is a good thing. I want to touch on this now so you guys kind of start to make these connections. So what is lactate? What's the big deal with lactate?
It's
What have
made
we
in the
heard
lip.
about it? So what have we heard about lactate?
So lactic acid buildup causing like DOMS basically.
Because there's something really important. Lactate equals lactic acid. I'm going to keep pushing your guys' brains to connect the dots. In acid, right? What is the definition of an acid in biochemistry?
Low
of
pH.
less than 7.
So it has to do with pH. So not necessarily that the pH is less than 7. That would be if a solution is acidic. But if we take a chemical that is an acid and we put that chemical into a solution, right? What's it gonna do? What's gonna happen? Let's say what's gonna happen to the pH if we add an acid into a solution?
It's
It's
gonna
gonna drop.
pH is gonna drop. Why? Why does pH drop?
the acid gives off a bunch of hydrogen.
Perfect. So the acid is our hydrogen donor. Remember? So we drop in the acid, and it donates hydrogen. So what happens is when we get lactate, and we get lactic acid buildup, we have an acid, and that's going to decrease the pH within the surrounding area. Do you guys remember what we talked about? What happens with our when our pH starts to get messed up, or if our pH goes outside of those normal ranges?
Proteins denature.
Right, we start getting issues with proteins. Maybe they don't denature immediately right away, but it is the further we get out of those ranges, that 7.35 to 7.45 pH value, right? The further we get out of there, proteins in our body doesn't work right. So that's one of the reasons if we don't have oxygen and we get lactic acid and all this buildup of lactic acid, the general area of those muscles that are giving off a bunch of lactic acid, pH decreases, proteins and everything that's going on there can't function quite as properly, and thus we get muscular fatigue. Because we're not producing enough ATP in order to continue to fuel the movement of the muscles. And also we're messing with the pH, which starts to mess with a little bit of the functions. And of course, once we stop moving and kind of get blood flow to the area, oxygen comes in, starts to bind everything, and we can start to clean out that lactic acid with our blood. We can clean out the carbon dioxide, all that kind of stuff. So that's going a little bit more into like the physiology side of stuff, but we tied it in to all the biochemistry stuff. Does all of that make sense?
Yes.
Yeah. So if we get kind of this idea of stuff, you guys will be miles ahead when you get to Phys 1 next trimester. Okay. So if an acid is our hydrogen donor, what's a base?
Hydrogen receiver.
Yeah, it's our hydrogen acceptor. It can also donate what?
Hydroxyl OH groups.
Yeah, our OH groups, because if it donates an OH group and an OH combines with an H, what do we get?
H2O.
H2O.
H2O, we get water, so it neutralizes it. And then that's going to help maintain our pH balance. Sweet. So we have all of our stuff with glycolysis. Perfect. We'll come back to that. Actually, we're going to continue with that. Because we have now pyruvate. So that was the if we don't have oxygen, right? In a state that we don't have oxygen, what's that called?
Anaerobic.
Yeah, anaerobic conditions. You just triple check. That's a word. Yeah. So this is our if we don't have oxygen, anaerobic. It's an easy word because literally aerobic, just a very quick difference there. So be careful when you're reading those questions. What about an aerobic condition? What happens to our pyruvate?
So then it goes into the Krebs cycle.
How does it go into the Krebs cycle?
Through alcohol.
Acetyl-CoA.
Yeah.
Yeah,
okay.
our pyruvate goes to acetyl-CoA. Does anybody remember the name of the enzyme that actually kind of
pyruvate dehydrogenase?
Yeah, we have a pyruvate dehydrogenases. I believe there are 3 of those.
Yeah.
I don't remember all the details on those. Let me look at the study guide, see if I have it. Geez, can I click every button except for the one that I want? Come on, computer. Come on. Sorry, it's trying to open up new tabs of stuff. There. Pyruvate dehydrogenase. I might have it as a picture and it doesn't pick it up. I don't have it. It might be in a picture of this somewhere in here. Yeah, we have those different pyruvate dehydrogenase enzymes that break it down and turn it into acetyl-CoA. Once it becomes acetyl-CoA, what happens from there? We remember that all this takes place in the cytosol. Where do we go next? What's the next process? What's the next step after glycolysis? What's our next process that we're going to go through?
Citric acid cycle.
Our citric acid cycle, aka our Krebs cycle. And where does the Krebs cycle take place?
Mitochondria.
What part of the mitochondria?
In the matrix.
Matrix.
In the matrix. So our glycolysis, we get the pyruvate, and our pyruvate is going to pass through the membranes and go into our matrix. So then we have the Krebs cycle. This is where you're going to probably have a handful of questions that are really just kind of like those raw memorization questions. With all of our Krebs cycle stuff. Let me pull up. I'm just going to pull this up real quick and go through the important things to remember. Where are you? There we are. So biggest things to remember in the Krebs cycle. It's in the matrix. The regulatory enzymes, the 3 enzymes we really want to remember that he's most likely going to ask questions about. The CIA is always watching. We have citrate synthase, isocitrate dehydrogenase, and the alpha ketoglutarate dehydrogenase complex. So the CIA is always watching is a way to remember that. There are 8 steps in the citric acid cycle. The whole process, each turn of the citric acid cycle, we're going to get 12 ATP per turn of the citric acid cycle. In total, from 1 glucose, we get 24 ATP. Because we get each turn, we get 3 NADH, 1 FADH, 2, and 1 GTP out of the citric acid cycle. The steps of the citric acid cycle, we see here, you don't need to remember. I believe, I'm pretty sure he doesn't ask these questions. You don't really need to remember like where like the H2Os and stuff come in and go out, or so much the exact type of reaction. Remember more the steps of it. So the mnemonic to remember this is couples cuddle insanely often after special sex for marriage obligations. And this is just one way to remember this, where if you remember all the different steps of the Krebs cycle. And then you're also going to want to remember where we have the NADH. So I am the most important. So I with isocitrate, am alpha ketoglutarate. The most malate is where we have the NADH coming out. Those are the main things you're going to want to remember from the whole Krebs cycle. Again, this is a lot of raw memorization. So I would go through this once or twice, get a general remembrance of kind of what goes on here. Don't spend a ton of time here just because it's not going to be, like, you already were tested on this. So you're not going to have a ton of questions pulled from this, but you'll probably have 2 or 3 at least pulled from something to do within this stuff of the Krebs cycle, whether that be the regulatory enzymes, kind of what is produced, where NADH comes out of, or like the different substrates of it. Okay. Let's go through the whole Krebs cycle, all this stuff. What actually is the end product of the Krebs cycle? What is it that we're actually trying to get out of the Krebs cycle?
ATP.
So eventually you want to get ATP, right? But within the Krebs cycle, what's after, as soon as we finish the cycle around the Krebs cycle, what is our actual immediate product? What do we have?
NADH.
Yeah, right. We have 3 NADHs and 1 FADH2 per turn of the Krebs cycle, right? So we have those. We have, come on, 3 NADH and 1 FADHP2 or FADH2. And we get 1 GTP, which acts similar to an ATP, so we count it as an ATP. Now from here, what's the next step? Where do we go from here?
Electron transport.
The electron transport chain. And where does the electron transport train located? Where is that located?
On the membrane.
On which
The
membrane?
on the
Yeah.
inner membrane space?
On the inner membrane. In the inner membrane, we have right here, these are also called cristae, the cristae of the mitochondria, the inner membrane. That is where our electron transport chain is going to take place, which is perfect because right now we have these NADHs and this FADH2 literally right there in the matrix to pass off perfectly onto that membrane to do what we need to do. But wait a sec. Over here in glycolysis, we said that we also made 2 NADH, right?
Mm-hmm.
But that's out there in the cytosol. So what do we do with that? How do we get that NADH to the inner membrane of the mitochondria?
Use the malate glutamate pathway. Is that it?
Very, very, very close. It's a shuttle.
Malate aspartate shuttle.
Yeah, the malate aspartate shuttle in all its beautiful glory. And remember, really, what we care about isn't so much like the NADH itself. We care about the electrons that the NADH is carrying. Because those electrons that are within that NADH, where that hydrogen is bonded to the NAD, that we have beautiful electrons that have a whole bunch of energy, and we want to use that energy to fuel our production of ATP. So we have our malate. The way that we get malate is that we take the hydrogen and those beautiful electrons away from the NADH. We put them onto an oxaloacetate, and it turns into malate. Malate can go freely across, being exchanged for an alpha ketoglutarate, and then it can break off, give it back to an NADH within inside of the mitochondria, and it converts back to oxaloacetate, and then it will interact. And then alpha ketoglutarate interacts on the other side to create glutamate, and then glutamate comes through. Glutamate reacts with the oxaloacetate to form aspartate and goes like through. So big things to remember here. Glutamate gets exchanged with aspartate, and malate gets exchanged with alpha ketoglutarate, and that is how the NADHs, how the electrons of the NADHs that were on the outside come to the inside in order to be used within the electron transport chain. And all of this stuff that's up here that we kind of skipped, this is all the stuff that we generally talked about. Just then we were just talking about it rather than reading through all of this. There's also something called the glycerophosphate shuttle. This is another way to get the NAD through. So you can use the malate aspartate shuttle or the glycerophosphate shuttle. Both of these will be working. Most commonly, he'll ask about the malate aspartate shuttle, but if he does ask about the glycerophosphate shuttle, biggest thing to remember here is that we use glycerol 3-phosphate and a flavoprotein dehydrogenase to take the electrons from an NADH, put it onto an FADH2, and that can go straight into the electron transport chain. Specifically, what complex within the electron transport chain accepts electrons from FADH2?
The succinate, dehydrogenase.
Which complex of the electron transport chain? Which complex protein?
Complex 2.
Correct, complex 2. Good job. And that leads us into our next chunk, the electron transport chain. So we have our NADHs, we have our FADH2s, all that good stuff. This membrane right here, which membrane is this?
What's the intermembranous space or intermembrane space?
So first, so let's go back real quick. I want to make sure this is super clear because I promise you, he's going to put a question or two on there about this. And if it's not super, super clear, you're going to get confused on it. So mitochondria, we have the outer membrane, the outside membrane. Immediately inside of the outer membrane, we have the intermembranous space. Outer membrane, intermembranous space. Then we have the inner membrane, where the electron transport chain is located, the inner membrane, and then the matrix. And actually, it might be... Let me double check something. Yeah. This is the intermembranous space. So we have the outer membrane, the intermembranous space, the inner membrane where electron transport chain is located, and then the mitochondrial matrix where the Krebs cycle occurs. Okay, make sure that that's super, super clear. So electron transport chain happens on the inner membrane of the mitochondria. So this is our inner membrane separating the matrix from the intermembranous space. Okay. This is going to be another one. You just need to remember, draw it a couple times and remember the order and the process at which this happens. NADHs give up their electrons to complex 1, and that uses some of that energy to cross hydrogen from the matrix up into the intermembranous space. Those electrons are then passed on to CoQ. Who remembers what are the other names for CoQ?
Ubiquinone.
Yeah. And what is CoQ? Is ubiquinone? What was the...
Coenzyme Q?
Yeah,
It's
coenzyme Q10.
It's not biotin, is it?
No.
Is that the other one?
No. It's coenzyme Q, aka ubiquinone. Sorry, I thought there was the Q technically stands for something else, but that's above our pay grade. We don't need to worry about that. So coenzyme Q. So it's coenzyme Q or ubiquinone, or it's CoQ or CoQ10. Sometimes he asks this question. CoQ is most directly related to what coenzyme? Making you think hard on this one. First off, what's a coenzyme?
It's a
Vitamin?
It's a vitamin. Coenzymes are vitamins. So what vitamin is most similar to CoQ?
Is it niacin?
That's it. Not niacin. It's a fat soluble. It's vitamin K. Vitamin K is the most similar to CoQ. He does have that question every now and then pop up. He does like that one. That also will come back in several classes. So remember that, because vitamin K, you need vitamin K to produce ubiquinone in order to maintain your electron transport chain. Okay, so we transferred our electrons to CoQ. We also mentioned this before. Our FADH2 gives its electrons to complex 2. And Antonio, you were right earlier when you said that succinate dehydrogenase. That's part of complex 2. So you were correct in saying that. The succinate dehydrogenase. Good job. I didn't remember that. So
No, trust me, man. Like, I studied the wrong material for the last exam.
It happens. It happens. So complex 2 also gives its electrons to CoQ. Super important. Remember, complex 1 and complex 2 do not interact with each other. They both give their electrons to CoQ. CoQ transports those electrons to complex 3, where it gives it to cytochrome B and then to cytochrome C1. And that also brings hydrogens from the matrix to where?
Intermembrane.
Yeah, the intermembranous space.
Intermembranous.
Across, what's this membrane called?
Inner membrane.
Yeah, the inner membrane. Good. Cytochrome C1 within complex 3 passes it to cytochrome C. Cytochrome C passes it on to complex 4, specifically to cytochrome A. Cytochrome A passes it to cytochrome A3. And then finally, it drops those electrons off after it's taken all of its energy, and it drops it off and lets it be oxidized into H2O. What happens next? Who can walk me through the process next of what's the deal with complex 5?
Three hydrogen atoms go through the ATP synthase, which produces 1 ATP.
Good, yeah. So what we've done, as we've pumped all of these hydrogen up here, right? We've used the energy of the electron to pump hydrogens up and across that inner membrane into this intermembranous space. And what we've created is a chemical or an electrochemical gradient. That's a fancy way of saying we got a bunch of stuff on one side, not as much stuff on the other side, and it wants to go from the high concentration to the low concentration. Your very first week in Phys 1, this is what you talk about, is concentration gradients and how they happen and how they move and how they react with each other. That's all week 1 of Phys 1. That and like 4 other things. So first week of Phys 1 hits like a truck. But right here, that's what we just made. We made an electrochemical gradient. Okay? Remember that word, gradient. So all this hydrogen that's up here, it wants to get back across because things in general don't like to be crowded. For example, if you take 100 people and stand them shoulder to shoulder and then put them into a massive warehouse, what are those people going to do? Are those people going to stay all bunched up, squished together, or are they going to walk away and get some space?
They're going to mosey around.
They're going to mosey around. They're going to get some space. What if you have a room jam-packed full of way too many people, and then you open these double doors? What do you think people are naturally going to do if you open double doors into a room from a room that's packed with people?
Move out of that room into the less packed room.
Yeah, they're going to move out of the room. They're going to go from an area of high concentration to an area of low concentration. So that pathway that it's able to follow is through the ATP synthetase, or through complex 5, which has ATP synthetase as part of it. And as they move through, they go through. Inside of the ATP synthetase is like a little wind turbine. And the movement of those hydrogens pushes that turbine and converts the chemical energy and the movement energy. So the hydrogen have a chemical potential energy because they have a gradient. And then they start moving. So the chemical energy turns into mechanical energy. And then it continues to spin, and it spins that little turbine. And then it takes that turbine and it uses it to bind phosphate to ATP, converting mechanical energy back into chemical energy to produce ATP. That was a little bit above our pay grade where we need to know for that. It eventually will come back up in other classes. That's why I talk about it like that. But big thing to remember, hydrogen goes through it, uses that energy to produce ATP. Sweet. Any questions, concerns up to this point about all the stuff that we've talked about?
Here.
Cool.
Yeah.
A couple more things. I don't think he has. Let's see if he has that's all of this stuff. That's all of this. Generating the protons. Little 1 mole of NADH produces about 2.5 moles of ATP. 1 FADH about 1.5 ATP. He'll ask a question like that. Just like, how many ATP are produced from 1 NADH? This is all still electron transport chain. Here we go. These guys. The uncouplers of oxidative phosphorylation. You guys should have seen a couple of these on your last test. Is that correct?
Yes.
Yeah. So those same guys, these guys, the rotenone, antimycin A, carbon monoxide or cyanide, all of these guys are blockers of the electron transport chain. Try and remember roughly where these block. Not really much else I can say there.
How I remember those?
Yeah, go for it.
All right, so rotenone. So like if you notice that they're ABC, right? So rotenone is outside of the alphabet, so it's H. It's between H and Q, so right there, and then B. So N is antimycin A is between B and C1, so like the alphabet, and then carbon monoxide is like right before oxygen. So that's how I memorize it.
Yeah. Rotenone, kind of outside between H and Q. Antimycin A, B, C. Cyanide or carbon monoxide right before oxygen. They prevent them by the electrons giving it to oxygen. Sweet. Good way to remember things. Thermogenin. What do we remember about thermogenin?
Hibernating brown bears.
What about them? What's
That
the purpose?
This is what they burn during their hibernation.
Yeah, so thermogenin is a natural uncoupler, right? So for what reason would we want to uncouple or undo or prevent like the actual formation of ATP during electron transport chain?
To save the stuff that we use to make it for when we actually need it.
So there's one reason. Look at the word thermo. When we think thermo, what do we think of?
Heat production.
Heat production, right? Thermo. Genin is a lot like Genesis. What does Genesis mean?
The beginning of.
Beginning or creation. So creation of heat, creation of temperature, right? So if we think brown fat, like hibernating animals, how much are hibernating animals using their muscles, moving around, jumping, running around, hunting, doing all that stuff?
Not very much.
Not very much. So do they have a need for constant production of ATP?
No.
No, right? But they do have a need for heat because it's really freaking cold out when they're hibernating. So what thermogenin does, it's a natural uncoupler, is it allows the whole process of the electron transport chain to occur. So it allows this whole process to occur, give those electrons to oxygen. But what it does is it actually uncouples it, takes apart or separates that ATP synthetase portion of complex 5. So that way the whole electron transport chain, glycolysis, the Krebs cycle, electron transport chain, all that stuff can occur. We don't get ATP out of it, but we do get heat production. So it's a way that the animal can use all of its resources, use that glucose and stuff that and the fat that it's stored up to create its own heat energy without just ramping up and having a ton of ATP. That way it's able to conserve its energy a lot better and just produce that heat and not have to do extra movement to burn off the ATP. Sweet. Any questions up to this point?
No.
In humans, what age of humans has more brown fat?
Babies.
David.
Yeah, the babies. That's why babies are so warm, even though they don't do anything besides eat and poop. Pathways of sugar metabolism. How do we feel about these pathways? We've gone over them a couple of times.
I feel pretty good about them.
We'll go through them pretty quick. If we have any questions, stop me and ask. Okay, so fructose. Fructose is that monosaccharide. Lots of fruits and stuff contain sucrose and fructose. Remember these are our disaccharides. The sucrose, lactose, mannose, those 3. Remember those. I promise you, you'll have a test question on that. I promise you will see that on boards. I promise you will see that in like 3 other classes. Just please remember those now. So sucrose is glucose and fructose. Elizabeth, what is lactose?
Glucose and galactose.
And what is mannose?
Glucose and glucose.
Yeah, I pick on you because I know that you know them at this point. Okay. So fructose. Fructose is okay. Fructose being converted to fructose 6-phosphate. Fructose has 2 different pathways that it can go through, right? It can go through the muscle or it can go through the liver. The liver is way more complicated and sucks, but it's fine. Muscle's easy. Literally the muscle, fructose goes up to the muscle. It gets phosphorylated by hexokinase. Fructose 6-phosphate. Fructose 6-phosphate is literally the second step of glycolysis. And so it just fits right into glycolysis, nice and pretty, and we're done. Hexokinase, right? There's another enzyme very similar to hexokinase that is found in the liver. What's that enzyme? That specifically one that participates in glycolysis. What's the name of that enzyme?
Fructokinase.
Yeah, good job, Leah. And Antonio. Okay, so that's within the muscle tissue. So now fructose goes to the liver. This one, a little bit different. Want to make sure you pay attention to this. It's not glucokinase or hexokinase in this case. It is a fructokinase within the liver. It's more specific. So in that, change it to fructose 1-phosphate. Our fructose 1-phosphate here is in the open chain form. And then we can convert to glyceraldehyde, and this we can go down either path, going either to glycerol to create lipids. Lipids, another word for kind of talking about lipids, or one of the most common lipids in our bodies. Remember which one I've talked about a couple of times? Anyone remember what that one's called? Starts with
Triglyceride.
Triglycerides. What that is a glycerol. That glyceride, triglyceride. It's a glycerol with 3 fatty acid chains attached to it. and that's what a triglyceride is. You'll learn all about triglycerides and lipid synthesis, lipid formation, and lipid breakdown. Kind of like this trimester, we talked a lot about protein buildup and breakdown and carbohydrate buildup and breakdown. Next try, it's a whole bunch of fat buildup and breakdown. So keep these little things in mind because these are going to come back next trimester. Okay, so we have the glycerol side. Go to glycerol 3-phosphate to DHAP, and then glycerol aldehyde 3-phosphate, and that's the intermediate into glycolysis. Or we can go straight from glyceraldehyde to glyceraldehyde 3-phosphate and into glycolysis. When you're studying, I'd take a moment just to read all these different enzymes. Take a moment as well. Make sure that you remember what the names of the enzymes do. It's like kinases. What do kinases do?
They phosphorylate.
Yeah, they add a phosphate group. What's the name of the enzyme that takes away a phosphate group?
Dehydrogenase.
Dehydrogenase are going to take away a singular hydrogen because it's dehydrogening.
That's a phosphatase.
Yeah, our phosphatase. So kinase adds on. Phosphatase removes a phosphate group. Dehydrogenase removes hydrogen. Isomerase. What does an isomerase do?
Rearranges.
Yeah, it rearranges the whole thing. What about a mutase?
It like moves one group to moves a.
Yeah, mutase is kind of also kind of a subgroup of isomerase, where it can rearrange a single. Usually a mutase like moves like one thing, like it flips one chiral center on one of these on a molecule. What about transferase? Elizabeth, this is the one you were just talking about, transferase.
Yeah, where it transfers one of the groups from one molecule to the other.
Yeah, what are the name of what's the name of that group? What kind of group is it?
Lost the word, I don't know.
A functional group?
Good, a functional group. Good
it.
job. It moves a functional group. Good. So just remember all these different names of the enzymes. If you know that name, that last part, kind of what it's supposed to be doing, that helps you a ton. Understand? Even if you don't necessarily remember the specific enzymes, if you remember generally what happens in the reaction, even like in general, and a lot of times the answers, like the question, will give away kind of what's happening, and then you can look down and pick your answer based off of that. So, and that helps a lot next try as well in Biochem 2, because you're gonna get hit with a ton of enzymes. There's a lot of them next try. There was a lot this try, but there's a lot next try too. Galactose. This is the one that we use. Remember, this is our UDP glucose. Galactose, we have galactokinase, which phosphates. UDP glucose and galactose 1-phosphate. The glucose and the galactose here, it swaps. So then we get UDP galactose, and it gives it the glucose. Then our beautiful UDP galactose 4 epimerase. Epimere, remember epimere is like that mirror image. So it changes it. Epimere is very similar to what we talked about with like the mutase with an isomerase as well. There's a lot that are like isomerases. Just kind of keep those names straight. And the epimerase converts the galactose to glucose. Glucose 1-phosphate gets phosphoglucomutase. Phosphate, glucose mutase. Talked about that mutase where it's going to be moving the different functional groups and rearranging stuff. glucose 6-phosphate, then can go directly into glycolysis. Mannose is hopefully one of the most straightforward. It gets acted on by hexokinase, and then the phosphomannose isomerase to go to fructose 6-phosphate, and that goes right into the Krebs cycle. Reviewing, what is the process by which fructose enters glycolysis in a muscle cell?
Hexokinase.
Good. Hexokinase converts fructose to what?
Fructose 6-phosphate.
Good. Fructose 6-phosphate. Good. Pentose phosphate pathway. What is the main job of the pentose phosphate pathway? What is one of the biggest jobs of the pentose phosphate pathway? There's two kind of really big ones.
Production of NADH.
Not quite NADH. Missing a letter there.
Oh, NADPH.
NADPH, because why is NADPH important? It's a super, super broad reason why it's important, not super specific. NADPH is used in a whole bunch of different biochemical reactions, biosynthesizing stuff, so making stuff. So we need NADPH to send it to other places to make more stuff that our body needs to be able to use to function properly. That's the one reason. What's the second big reason?
Makes ribose.
Yeah, it makes the ribophosphates. Why do we need those guys?
Protein synthesis.
Eventually protein synthesis kind of later on. More so than proteins.
Like nucleotides?
We use them for the nucleotides, are nucleic acids. So very, very important thing. Proteins and nucleic acids are different from each other. Nucleic acids, what's one example of a nucleic acid?
So like adenine.
Serine.
That's like a specific adenine is a specific one. Think bigger once they're fully formed.
DNA.
Nucleic acid, yeah, DNA, RNA, messenger RNA. Those are our nucleic acids. So the ribose, deoxyribonucleic acid. So nucleic acid is our DNA, our RNA, our mRNA, those guys. Proteins are buildup of just amino acids. And the way that we build proteins is that we read the DNA, we read the structure of the nucleic acid to then pair it. And then, that's the whole process of building mRNA. And then the mRNA goes off, mRNA goes and gets read by the tRNA, and you have the S40 and the S60 ribosome come and they bind, and then the tRNA comes, and then you get the shifting and bringing in more amino acids and creation of a polypeptide chain, and then that gets all folded beautifully, et cetera, et cetera, et cetera, and then that creates a protein. So in the grand scheme of things, nucleic acids are used to make proteins, but specifically ribose is used to create nucleic acids, DNA and RNA and those good stuff. We have NADPH. We talked about NADPH for synthetic purposes. Create fatty acids, sterols. You'll learn about these guys more next trimester. Pentose phosphate pathway comes right back up in Biochem 2. So don't let this just slip out of your brain. Keep that in your brain. So producing the NADPH, we have two different phases. We have the oxidative phase of PPP and the non-oxidative phase. Within the oxidative phase, Let me find there should be a nice picture. Here we go. Look at that. I added all this stuff in there for us. Glucose 6-phosphate gets acted on by our enzymes to go to 6-phosphogluconate, and an NADPH is formed. Then we go to ribulose 5-phosphate, and we produce another NADPH. That right there is our oxidative phase. Ribulose 5-phosphate. Is ribulose 5-phosphate, is that a ketone or an aldehyde?
I'm guessing aldehyde.
50-50? You got it wrong, but that's okay. Ribulose is going to be our ketone, and it gets converted to ribose 5-phosphate, which is our aldehyde. Ribulose 5-phosphate can also be converted to xylulose, which is another ketone. So it can convert it to either, but we typically talk about ribulose 5-phosphate being converted to ribose 5-phosphate. And ribose 5-phosphate is what goes and becomes that nucleotide in DNA and RNA. And if, let's say, we don't need to be creating DNA, RNA in this case, the ribose and the xylulose can get acted on by other enzymes called a transketolase or a transaldolase. Transketolase acts on, which of these do you think that one acts on? The xylulose or the ribose?
The xylulose.
Yeah, because xylulose is a ketone. Transaldolase acts on the ribose because ribose is an aldehyde. I'm saying that right. Correct, right? The
only way I remember it is if it ends in lose, it's a ketone, and if it doesn't end in lose and it's ose, then it's an
aldehyde.
Aldose.
Yeah.
Aldehyde.
Aldose. Is it aldose or aldehyde? My brain. Now that I'm saying it out loud, it's one of the two. It's fine. I think it's supposed to be an aldo and aldose, whatever. Know that these ones are ketones. The other one is not a ketone, and that'll be what you need for answer choices for the test. Anyways, these can get converted back into fructose and glyceraldehyde and get put back into glycolysis if we need them. That's when we don't need the ribose. When we do need it, then it continues down. Okay. Those are our big kind of pathways and stuff. How's everybody feeling? How's everybody doing? Kind of just powered through a lot of information.
This is all the information I'm feeling okay about, but
yeah,
that's good.
It's like the early information that I'm worried about.
Do we want to jump back to the very beginning?
Yes.
I mean, whatever everybody else wants to do, but that's what I'm more worried about.
Drake, I can go over the last 3 chapters that we from biochem.
What do you mean the last 2 chapters?
So the last 3 chapters are over. Hold on, let me pull up my notes.
Oh, are you talking about
translation, synthesis? What is it? Translation, and then RNA structure and structure of nucleic acids and DNA replication.
I not have any of that on this study guide?
Oh, I think so. They're at the very end, but I'm just having trouble understanding it.
Yeah. To be honest, the best way to understand all that stuff, because it's another pathway, it's another process. Let me just do this. Let me pull up. Biochem lectures.
You have it in
Actually-
your final exam study guide, Drake.
Was I just in the wrong one?
I think
Let
you
me
may
see.
have just been in exam 3 because it's in your final exam 1. I'm looking at it right now.
Ah. Foiled again. Let's see. Bam. Let's see. Transcription. Here we go. This stuff, right?
Yeah.
Okay. Definitions. Big help. Know the definitions. These will be questions on the test. There were a handful in the Linus Learning. Antonio, I don't know if you, or Aaliyah, I don't know if you saw it. I did post the recording of the Linus questions from last week that we did. That covers all of this stuff as well. So you can go back and watch those on the YouTube channel. And
Yeah, I
see
saw
all
it.
those.
Thank you.
Okay.
I just took a cell biology course, and this was a good like 4 weeks of it in my undergrad. So it's pretty fresh in my brain still, thank God.
Oh yeah,
Look at you.
they beat it into you. Okay. Replication. DNA makes DNA. This is replicate. It's what kind of replication? What's the name? How it gets replicated?
Is that the like semi-conservative?
Yeah, semi-conservative. What does semi-conservative mean?
Like it keeps the original part of the original strand as it continues to replicate.
Yeah, it keeps one of the parent strands as it makes the daughter strand. Transcription is DNA making RNA. So DNA specifically, it makes mRNA. What does mRNA stand for? What is the M?
Messenger RNA.
Yeah, messenger RNA. And then translation, where the mRNA gets read in order to form the polypeptide chain, which then makes the proteins. How do we feel about like chemical structure stuff, like purines, pyrimidines, all of this stuff? Is this okay? Can we skip this?
Yeah, I'm good
Yeah.
on it.
Okay. Keep going. Our little chart here. Nucleoside, nucleotide, nucleic acid. We talked about this a little bit last time. And also there was another one before, just the base. Wasn't there another name? Nucleoside, nucleotide, and there was another nucleo something. Do you even remember? I thought last week we talked about that.
Because we were trying to figure out. Oh, I think it was just those 3. I was trying to figure out why adenylate was the nucleotide in that question because I hadn't looked at the differences.
Yeah. Okay. Nucleoside is the base plus the sugar. Nucleotide is the base sugar plus the phosphate. So look at the differences here. Nucleoside ends in -ene, adenosine, sine, all of these, -ine. The nucleotide ends in 8. So this is all about the building structure. The phosphodiester bonds is what connects the ribose parts of all of our stuff. Still feeling okay through all this?
Mm-hmm.
Okay, we'll get into like the actual stuff. So the hydrogen bonding between A and T and G and C. You have trouble remembering that? It's at the golf course. A goes to T, G goes to C, in RNA, U replaces T. He'll usually ask a question about this, Chargaff's rule. That's just saying that the total number of A and T, the total number of A is equal to T, and the total number of G is equal to C. It's because
Did not see that
they're
last
paired.
trimester.
What was that?
Didn't see that one last trimester.
Okay. He might not ask a question about it. He might ask it like not specifically like Chargaff's rule, but he might ask you about like there were a handful of questions in Linus that were like, if you have like the structure is this percentage of G and C, what percentage of the strand of DNA is made up of T? So like simple math stuff like that. Like it's like addition and subtraction stuff. The structures of DNA, you can have B, A, and Z DNA. Remember that B DNA is the most common. This is what our DNA is because it's right-handed. Right-handed is the most thermodynamically stable. Denaturation and renaturation of DNA. We should know that stuff. Denaturation, it's the same thing as proteins, breaking it down. Alkali, heat. And then once you remove those things, it will come back together. Okay, DNA replication. Antonio, is there anything specific? Do you feel okay with DNA replication, or do you want to start a DNA replication?
I'm okay with DNA replication.
Okay, what part specifically are you more kind of like? What?
Let's see. It was like the enzymes, like the DNA polymerase and all
These guys?
that. Yeah.
Just kind of what the function are of them.
Mm-hmm. Yeah.
Okay, so this is gonna like kind of tie in with each of the different steps. So for example, DNA polymerase, it's the enzyme that creates the DNA molecules by assembling the nucleotides. So when we get this creation of the leading strand or the lagging strands, the DNA polymerase is what's going to bind and actually go down, and it's actually going to be the protein that is building the new strand of DNA. So that's its job.
Okay.
RNA polymerase does the same thing as DNA polymerase, except it builds RNA instead of DNA.
Okay.
The DNA ligase, once we produce the Okazaki fragments on the lagging strand, ligase is gonna come in and glue that stuff together. Yeah. Topoisomerase. I start with helicase. Helicase is what unwinds the DNA, so it breaking it apart here and kind of starting to uncurl it right here, so that way it can continue to open as we produce the two new strands. Topoisomerase. I explained this last week. Like when you take a string or a rope that has multiple parts in it, you start to pull this and separate the two mini coils of rope or whatever, or string that make up the bigger coil.
Yeah.
So do I have I think I should have a string here somewhere.
Okay, so yeah, like basically you just start to unwind them, like the little individual strands.
Yeah, if you start unwinding both strands, if you pull it pretty quick, you get like a bunched chunk at the very bottom, right?
Yeah.
Where it's like starting to separate. So that's kind of what starts to happen with DNA. And that's called supercoiling. So to prevent that, we have DNA topoisomerase, which it prevents that supercoiling. So it's going to kind of, as our helicase pulls and breaks it apart, this topoisomerase is going to be down here where that like intense coiling is happening. And it's going to kind of break the DNA strand to release the pressure and then reattach it. Break it, let it release the pressure and reattach it over and over and over again. So that all has to do with DNA replication and RNA formation, mRNA formation, right? Our exonuclease and our endonuclease. These guys are responsible for if we have mutations that occur. Did he spend a lot of time on mutations with you guys? Going over any of those?
Not that I recall in ours.
I mean, maybe briefly, but not really that I can recall either.
I don't have it on my study guide at all, so I guess there must be another. There must be another class in the future that goes into this stuff.
He kind of goes over it, like the different types of mutations.
Is it in the book? I feel like if you touch that, because I don't remember seeing like a single. There might have been like one question about like a point mutation in the Linus stuff.
Yeah, there was one point mutation question. That was it.
He probably won't hit it super hard. Those, I think later on, you'll talk more about those as well. There's a couple of them. There's like, I would double check types of DNA mutations. Like there's a decent chunk of them. You might recognize some of these: nonsense mutation, missense, frame shift, substitution, duplication, deletion. Those are the big ones. Like a deletion is exactly what you sounds like. One of the base pairs gets deleted. A substitution, one of them gets changed out. You have a frame shift mutation, which means you have 3 base pairs get added or taken away. It's an insertion or deletion of 3 base pairs. If you think further on down the road, that's going to cause an extra amino acid to be put in right there. Sometimes that frame shift mutation can really mess up the protein that it's supposed trying to be building. The nonsense mutation, meaning it changes, so it's a point mutation, so one of the individual base pairs gets changed so that it turns into a stop codon, and so it ends up stopping the polypeptide chain formation early. Nonsense mutation is, if I remember correctly, is one of the big reasons why people become lactose intolerant. You get a nonsense mutation that prevents the lactase enzyme from being produced by the body. Sweet. And that being said, exonucleases and endonucleases are the enzymes that are designed to fix those mutations. So the exonuclease, yes, they can kind of work together. So either the exonuclease is going to start either at the 5' or the 3' end and work its way back towards the mutation, or if the endonuclease can go right along and directly to it and snip it out of the middle without having to break for either end. And if the exonuclease, let's say exonuclease comes, or the endonuclease comes and snips it open, the exonuclease can then come in and work from that opening as well. It's more, the only big thing you need to remember about exonuclease and endonuclease is just where they start. Exonuclease, exo meaning outside, they start from the outside, the ends of the strand of the DNA. The endonuclease starts in the middle of the strand of the DNA to fix mutations. Transcription and translation. Big one you want to remember is the RNA polymerase 2, that it synthesizes the mRNA precursors. He likes that question. The different steps, just know the definitions, roughly what's happening in each of these. Initiation, where you have a promoter. There's a specific DNA sequence that's called the promoter, also known as the cat box or a tata box. He doesn't really ask questions about these. These are a little bit more specific. Just recognize the name, cat and tata, talking about it being a promoter. That's the way he's going to ask that kind of question if he does ask it. That's a little bit above kind of what we really get into. Are we okay? Are we okay with this part of synthesis, or do you want me to go over it deeper?
No, you're good.
Okay. Remember introns and exons, which ones are the coding or non-coding regions. He'll have at least 1 or 2 questions specifically about that. Just super, super simple. Give me points. It's literally a 50-50 shot of getting the correct answer. Just remember which one is which. Translation. He's not as mean to you guys. You guys don't really need to memorize all these. The ones that I would know are AUG. He had that question on Linus like 12 times. What is AUG code for?
Methionine.
Methionine. Know that one. And you might want to know the stop codons as well, maybe. But I don't think he's not looking for you to just memorize this whole chart, so I wouldn't worry too much about that. Just know your start codon AUG. And if you can remember the stop codons, that's great. If not, you're probably going to be fine. All of this, just know where stuff is, where the anticodons are, where the codons are, because those will be questions. Pretty straightforward. He'll probably put them in more words, but those are going to be the questions. I think in his stuff he uses 40 and 60S, 30S, 50S, 40S, and 60S. It's the same thing. It's fine. Again, this is just the whole process. I highly recommend, if you get confused about any of this stuff, just look up a YouTube video. Like literally do translation step by step, translation of proteins step by step, and there's phenomenal like 3 to 5 minute YouTube videos that show this in action and show it way, way, way better than I can explain it. So go to the almighty internet for this sort of process, and you can just look up like simple protein translation videos, something like this, and it'll walk you through kind of step by step exactly what's happening. And this is it should, let's see if this one does it. Yeah, it shows you kind of how it goes in, and then you can pause and look at this. Remember this, we have our P site, our E site, and our A site. Does that sound familiar? P site's where it's conjoining, E site where it's going to be exiting, A site is the active site where it's coming in to join in.
Yep.
All of this stuff, E, P, and A. All of these steps. And then just a couple of the inhibitors. He asks the questions of Linus. A couple of these. He asks about a couple of these inhibitors. So review the Linus questions. I guarantee a lot of those exact same questions are going to pop up on the final. Okay. Any other specific questions? What's the lecture? How are we doing? We want to keep running through stuff, go and start from the bottom? Start from the beginning?
I think we can start from some of the stuff we probably need to refresh on,
Yeah.
like the
Okay.
basics.
Honestly,
Yeah.
I definitely need to go over some of that.
Again, and if you want to remember, I do have on the YouTube channel, I have all of the like the chemistry review, all that sort of stuff. You can pop it in. If you can handle this, some people's brains cannot and do not do well with this. One of the ways that I would study is I would put on either lecture videos or something of whatever the topic was that I was reviewing, and I would put it on in the background. So that way, like, I could half listen to it. So that way, like, I would be working on the study guide, working through the study guide. And then you can listen to the recordings at a little bit increased speed. And kind of, as you listen, be working on this, Googling stuff. And every time if you hear something in the lecture that you're like, pause and like, wait, what was that about? Then you can quickly go back to the lecture and listen to it. It is a technique that can work with certain classes if you're able to do it. If you have high like anxiety, OCD kind of stuff, might not be great if you're like sensitive to a lot of stimulation. But for me being king of ADHD, I really liked having like a lot of stuff going on at once. So
That's gonna require that I turn the baseball game in the background off, though.
which, you know, it might be worth it if, you know, when it comes when it comes to crunch time. Okay, starting from the very beginning, he probably won't ask a lot of questions from this kind of stuff, just because there's a lot of like gimme opportunities in the new material. But you have your cations, you have your anions. The cations are positive, anions are negative. Know your bonding, ionic bonding, covalent bonding, nonpolar bonding. When we talk about building peptide chains, what are the bonds called between peptides in a polypeptide chain?
Covalent.
They are covalent. What's the other name for them now?
Hydrogen?
Peptide bonds.
Oh, peptide bonds.
In the polypeptide chain, do you remember what step of protein synthesis the polypeptide chain is?
Step 1.
It is step 1, which is called what? What structure?
Primary.
Primary structure. Good. Primary structure. Is this kind of stuff tickling the brain cells a little bit in remembrance?
A little bit.
Primary is those polypeptide chains, and then you have secondary structure where you have alpha helices and beta something else. What are the other something? The beta pleated sheets. The beta pleated sheets, you can have parallel or anti-parallel. Yeah, there's a lot of stuff in there.
Yeah, there is.
Yeah.
That's all the stuff I don't remember.
Do you remember what type of bonds are typically used in the secondary stage of protein synthesis?
Hydrogen.
Hydrogen. What about tertiary structure?
Covalent.
Yeah, like disulfide and van der Waals.
Tertiary is going to be some disulfide linkages, but also hydrogen bonding as well. And then quaternary as well. You're going to have the van der Waals forces and hydrogen bonding. If you don't remember, guess hydrogen bonding. 90% of the time, you're right. So, okay. Non-polar covalent bonds, polar covalent. Do you remember all this stuff? Polar versus non-polar?
Yes.
Yeah.
Yeah.
Hydrogen bonding. We love hydrogen bonding. Hydrophobic versus hydrophilic.
Yep.
Good.
Yeah.
What's another way of saying hydrophobic? We say,
Lipophilic.
yeah,
Oh.
lipophilic or lipophobic for hydrophilic. Good. He makes a big deal. He would always say, I don't know if he said this to you guys, like dissolves like. Remember hearing him say that?
Yeah.
I remember that from high school.
Yeah.
I
So
remember
if
that
you
from
have like,
somewhere, but I don't know where.
so kind of like, polar solutions will dissolve polar compounds. Non-polar will dissolve polar. Hydrophobic stuff will stick with hydrophobic. Hydrophilic will stick with hydrophilic stuff. So tonicity, hypertonic, hypotonic, and isotonic solutions. What happens to the cells? I always remember hypo sounds like hippo. Hypotonic solutions, you get expansion because hippos are chunky. And if you remember one of them, you should be able to remember the other. Acids. What is an acid? What does it do with the protons, with the hydrogens?
Donate.
Donates them. Weak and strong acids. What's the difference between a weak and a strong acid?
A weak acid is only gonna like partially donate its hydrogens, where strong is gonna give all of them.
Good. So if a weak acid gets put into a solution, does the pH change a lot or a little?
Just a little.
A little.
Just a little bit. What about if I put a strong base into a solution?
It's gonna take all the hydrogens.
Yes. What happens to the pH?
It
Gonna
goes up.
change a bunch.
It goes up.
Yeah, it's gonna go
By a
up
lot.
by a lot. Yeah. A buffer. Does anybody remember what a buffer is?
Oh.
No.
It's
Weak acid
ahead.
and its conjugate base. So a weak acid is one, it's not going to fully dissolve. The conjugate base is going to be whatever that is that you put in without its hydrogen. So here, AH, here's our weak acid. And then once the hydrogen comes off, that is our conjugate base. And what that's going to do, and this is double arrows because it's going to go back and forth and it's going to hit an equilibrium point. Because as a weak acid, it's going to give up its hydrogen, but kind of reluctantly. So it's willing to give up its hydrogen, but it's also willing to grab it again. So the buffer systems are something that we can do to help manage the pH, easily keep it balanced, which is why within our body, we have stuff like the bicarbonate buffer, which is going to come back next try and the try after that and the try after that in a couple of different classes. You don't have to memorize the whole thing super intensely. It comes back. Just look out for it later on. But the main reason for the buffer is so that you can closely monitor the pH and make sure that pH stays appropriate. Because what's the appropriate pH for our bodies?
7.3435
to 7.35 to 7.45. Good job. Buffers. We talked all about this stuff. pK values. This is the strength of the acids. The more negative it is, the stronger the acid. That's about as much as he'll ask you about pK. Like he'll give you like the pK value is negative 15. Is it a strong acid or is it a weak acid? If it's very, very low negative number, it's a strong acid. All about pH. We talked about that. Henderson Hasselbach. All this stuff bringing up the trauma from the beginning of the try.
Yes.
Yeah.
Yes.
A little bit.
I hated this stuff. And then like the PKA bull crap, all of this. I hated this stuff. I still hate this stuff. Let me find. Here we go.
Hold on.
Remember this stuff?
Yeah.
VMAX and KM and
Yeah, I still don't get all that.
some of this will come back. I'm still this is probably a good thing. I don't want to say don't study it. But like you might get one question on this stuff. Maybe two for the whole test. And that's like it's a big but.
I'll be honest, I've just taken an L on those.
Yeah,
Taken
like
an L.
Yep.
this might be something if you guys want, like this is something that we can dive into like over the break if you really want to. This will come back in try 4 with Dr. Perryman's class. No, try 5
What class is
with
that?
pharmacology and toxicology.
Oh.
But
Okay.
I feel like Dr. Perryman will explain it better because I've had him before and I like the way that he teaches better. So
Yeah, but to be completely honest, this stuff just doesn't make sense. Like straight up, just it does, but it doesn't. Like Ninja Nerd has a really good video that he dives into all of this, like how these equations are derived and why these things are what they are. It's like an hour long of like
My timer.
pretty heavy math. So I watched it. I enjoyed it, but I still like I'm more like this stuff. Memorize this and call it a day. Competitive inhibitors increase the KM. You have decreased affinity. Non-competitive inhibitors, no change in KM, decrease the Vmax. Like that's really all you need to know. So yeah, do with that what you will. Okay. Oil rig, oxidation reduction reactions. Please, if you do not understand oxidation reduction reactions, learn them before the test. Learn them before next try. Learn them before several tries after this, because this will not go away. It really won't. This is going to come back a lot in like physiology type stuff, in understanding like nutrition, all of that kind of stuff. Like you need to understand oxidation reduction stuff.
The second I stepped foot into undergrad, it started haunting. So it's not a lie.
Oh yeah. Oxidized and reduced forms of molecules. Here we go. I feel like he has a couple of questions usually like this. This is the stuff he really likes, these kind of questions. If you go on Linus, do those practice questions, and here just understand like primary to aldehydes, secondary alcohols to ketones, aldehydes to carboxylic acid, and that's literally how the question is going to be. It's like primary alcohols are oxidized to which of the following? Aldehydes. Aldehydes are reduced to which of the following? Primary alcohols. Like that's how he's going to ask these questions. And he usually will throw one of these in there. He loves this stuff. He loves his aldehydes. He loves his ketones. Functional groups. this is another thing. Don't go back through and try to memorize all of these. Hopefully by this point you've seen enough of like the common ones, like the methyl and the ester and the amino and the disulfide and the phosphoryl. Hopefully you've seen them enough times to kind of remember the hydroxyl. Again, hopefully you've seen them enough times to kind of remember some of them. He doesn't usually ask like these specific questions on the final exam though. Like which of these is this functional group? Amino acids, the chiral carbon, zwitterion. I feel like he might have asked a question about zwitterions on the final last time, but not like this stuff isn't super deep. Peptide bonds, the amino acids. He'll probably throw in a question or two about amino acids by their polarity or something. So like, usually he'll have like the sulfur containing, the branch chain. Remember sulfur containing. So to be an MCC, you have to have sol. And this is cysteine, methionine cysteine, technically cysteine. These ones are like kind of, but not really. Live on a branch for the branch chain. The acidic amino acids are acidity of as and glutes, aspartic acid or aspartate and glutamate. Yeah, don't spend a ton of time on these. Like this is another one where like if he has a question, like one question on it, know these, the MCC, live on a branch, acidity of as and glutes, because those are the most common ones he's gonna ask about. Or like, and other than that, like it's just not worth the time to answer that one question if he asks it specifically. Aromatic rings are your pal Tyrone trips you in the ring. Phenylalanine, tyrosine, and tryptophan in the ring structure. Disulfide bonds, cysteine. This will come back. Disulfide bonds will come back a lot when we talk about insulin. You'll talk about insulin in Biochem 2, and you'll talk more about insulin in Biochem 2 than a lot in Phys 2 and Tri 3. Modification of amino acids. This stuff isn't crazy important. Some of these stuff you should know. Acetylation, carboxylation, hydroxylation, oxidation. Structure, function, relationship in proteins. You have the fibrous. Think like grass. It's insoluble. It doesn't dissolve in water. Globular proteins. I always think of like slime. If you put slime in water, it's going to dissolve. Primary, secondary, tertiary, quaternary structures. All of these good stuff. Alpha helixes, the beta pleated sheets. The bonding. The right-handed alpha helix is more stable. That's why our DNA is what kind of DNA?
B, right-handed.
Yep, our B, right-handed. Who are Watson and Crick's?
They're the ones that like found
The
structure of the.
Yeah, they're the first one to map and present that double helix DNA structure. Proline and glycine. This one will probably come back. Proline and glycine. Oops. Boop. The heck? There it is. Proline and glycine, because glycine is super, super small. What's funky about glycine that's different than all the other amino acids? What's special about glycine?
Non-chiral.
Yeah, it's the only one that's non-chiral. And because it's so small, it helps. It can move with proline to help with the beta pleated sheets have a nice fold. The bonding, hydrogen bonds, hydrogen disulfide linkage for tertiary. And then quaternary is the van der Waals forces, hydrogen bonding, blah, 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 blah. This is the only time that like van der Waals forces will show up. He's had like, remember he had like one question through all of the tests about van der Waals forces. So denaturation of proteins, we've been over that. What causes that heat, pH, detergents, alcohols, this kind of stuff. Anything that's super intense. Oxygen binding, enzymes. So a lot of this stuff at the beginning, hopefully as we've gone through some of this, you can see like I've tried to tie a lot of this back as we've gone through tutoring. So a lot of this like should look familiar, like we've talked about it. If you watch the tutoring stuff, we've talked about a lot of this kind of over and over and over again. Kind of what enzymes do, the cofactors versus coenzymes. I know, Elizabeth, I've beaten that into you pretty good.
Yeah.
We're cofactors. I remember fear factor. You have to be pretty metal to go on fear factor. Our cofactors are metal ions that help with reactions. Coenzymes are our bigger stuff, like our vitamins. All of our B vitamins, the tall, rich nudists played pickleball for centuries. Thiamine, riboflavin, niacin. The 2Ps. Pyridoxine. And the other one is pyrimidine something or other. Biotin, folate, and what's the last one called?
Cobalt.
Yeah, no, not cobalt.
Hmm?
Cobalamin. Cobalamin?
Yeah, cobalt. Yeah.
Yeah, because cobalt's a metal. B12. Yeah, cobalamin. Cobalamin. Which makes me think of, I don't know if any of you played Pokemon. Cobalamin reminds me of this Pokemon. Don't know why. It just does. Enzyme classifications. I've beat this down your guys' throats as well. All of our different enzymes, right? Transferase, hydrolase, lyase, isomerase, ligase, like what these enzymes do. That's why I've done that so much. The kinases, the phosphatases, dehydrogenases, all of those. That's why I've done that over and over and over again so that you guys remember all of these different types of enzymes. Generally, how the enzymes work, they lower the activation energy. Things that affect enzyme activity, pH, temperature, the substrate concentration, activators, inhibitors. Here's that competitive versus uncompetitive or non-competitive stuff. Literally just remember these. The Vmax decreases or Km decreases. Max decreases or Km is unchanged. That'll get you where you need to be for these test questions. The Michaelis-Menten equation probably won't pop up. It's kind of like more within the unit and then it goes away. Allosteric effect. Do you remember what we talked about with allosteric binding? What that means?
But it's where the inhibitor or whatever else, like something else comes and binds onto the enzyme at a site other than the substrate's site.
Yeah,
Other
other
than
than
the
that
active
active
site.
site. Yeah,
Yeah.
perfect. That allosteric binding. So when we talk about like allosteric binding as well, We can also think of stuff like negative feedback loops. Who can describe to me what a negative feedback loop is?
The production of something stops the production of it once there's enough.
Perfect. And that part of that is because some of those products go back and act as like allosteric, or like some of the products or portions of the products or things that are produced are going to go back and allosterically inhibit and stop enzymes from continuing production. So you get some allosteric effect depending on the reaction. All the reactions are different. You don't need to know like specifically what allosteric reactions occur, but knowing what it is and get us associated with that feedback loop. Titration, don't worry about that. G protein coupled receptors. Some of this stuff will come back, remembering the hormones, like the steroid hormones, the thyroid hormones, the transmitters. Remember the kind of receptors that they bind onto. For example, acetylcholine, norepinephrine, those guys that they have the plasma membrane receptors. Remembering roughly kind of what things bind to G protein coupled receptors. Steroid hormones, thyroid hormones. Where do those bind in a cell?
Nucleus?
Yeah, they're nuclear receptors. Yeah, they have nuclear receptors. If I throw out the word tyrosine kinase, what binds to a tyrosine kinase? Yeah, insulin. Remember that one. That one comes back in future classes. Big ones, remember steroids, thyroid hormone. He loves thyroid hormone questions. He'll ask you, I guarantee there's a thyroid hormone question. Probably super straightforward. Where does the thyroid hormone bind? And it's going to be a nuclear receptor. Insulin and glucagon, the relationship between those two. Remember, insulin is designed to bring glucose from the blood into the cell. It lowers blood sugar. Glucagon raises blood sugar. Where is most of our glucose stored in the body?
Liver.
In the liver. Which is responsible for maintaining blood sugar? The liver or the muscles?
The liver.
The liver. Why?
Because it secretes insulin and glucagon.
The muscles can't break down glucose anyway?
The muscles can break down glucose and they can break down glycogen, right?
Mm-hmm.
But they can't release it. Once glucose has gone into the muscle, it can't release it back into the blood. Whereas the liver can do that. It can unphosphorylate the glucose and release it back into the bloodstream. When we're talking about insulin and glucagon, what's the organ that produces that insulin and glucagon?
Pancreas.
Pancreas. Specifically, where in the pancreas?
Islets of Langerhans.
Yeah, and specifically within the islets of Langerhans, what type of cells produce insulin?
Beta cells.
Yeah, what about glucagon?
Alpha.
Oh, good. And then later on, eventually in a future course, you'll learn about delta cells. And delta cells help secrete another hormone called somatostatin, which then you'll learn all about like pygmies and how they don't have that specific hormone and how it correlates to like growth hormone and how growth hormone is connected to insulin and glucagon. But that's later on in the future, so don't worry about that right now. So remember, focus on insulin and glucagon and those guys. Do those nuclear receptors? Remember the cascade. I just saw the word. The cascade pathway. When we talk about a cascade pathway, what is that referring to? It's a
How a
cascade pathway.
ligand produces its effects.
Yeah. So you have the primary messenger, the first initial ligand, and it can go into a secondary messenger. This is kind of like the domino effect, almost like a pyramid, like domino effect, where you have one connection and then it sends out a message and that recruits two messengers and that recruits each of those recruit two and they recruit two each and so on. And so it creates a cascade of response. So like remember this chart, this is a chart that's in the book. These are plasma membrane receptors, these are nuclear receptors at testosterone, the thyroid hormones. Our G protein and protein kinase A, all of this, our G protein coupled reaction. Does this ring a bell? All of this stuff?
Yes.
You'll have a handful of questions on this, probably just one or two on this whole process. Will be thrown in there, something around it. So.
Yeah, this process is where I had my aha moment. And so like everything else past this, I started to get. Everything before it, nope.
Good. Well, it's good you know where. But yeah, like, and kind of going to the previous stuff before this, going back to it, especially like when it's a little bit organized, hopefully it doesn't feel quite as bad as when you were first doing it, hopefully.
Yes.
So
definitely better just like from bringing it up in like our tutoring sessions, like, oh, okay, yeah, now I remember that. Like,
yeah. So a lot of this is going to be from here on out. We're going to go ahead and end for today, but do the practice questions on Linus. And that's going to be key, is every single time you get one of those practice questions wrong, look at the answer choice. Don't just like, oh, that's the answer and then move on. Stop for a second. Every time that you get one wrong, look at it and be like, okay, why did I get this wrong? And what am I missing? What's the disconnect? And as you keep identifying those things and going through more and more questions, you'll start to see like, okay, I'm really like, you find where your deficits are and you're able to tackle just those deficits in order to go through everything. And it's especially helpful like if you don't need to like 100% ace this test, like as you go through, you're like, okay, you start to feel more comfortable with all of the material.
Well,
Okay,
you,
you guys. Thanks you guys for coming in, for joining. Hopefully we learn a little bit of stuff. Have a great rest of your day. And also, I'm around like all Thursday. So if you guys have any questions, concerns, anything going on, or if you have like questions and want to review stuff for a different class, let me know. I have Thursday off of CBI and actually just found out that my morning meeting on Thursday is asynchronous, and so I don't have that meeting. So I'm available pretty early in the morning.
How are you with FBI?
Better than I used to be when I took the course. So if you have stuff you want to review, and if I don't know it off the top of my head, or I can't like figure out like right away, like we can sit down and like work through stuff. So
okay,
just let me know. Send me a message either on GroupMe.
That'd be awesome, helpful, because I'm struggling in her class, kind of.
Okay.
I'm right on a border of B, C, and I kind of want to stay towards that B.
Gotcha. Just shoot me a message and let me know kind of what your schedule is, and we can figure something out.
All right, thank you.
Guys. Have
thanks,
a good night.
You too, thank you.